Okay, so now in video 2, we're going to talk about what central pattern generators are and how they work. So in the locust nervous system, they have a brain, and that's pictured in figure B, in part B of the figure here at the top. And then there's a ventral nerve cord that connects kind of all of the different central nervous system structures along the length of the locus. And then there are ganglia, which are clusters of neurons that are basically like mini brains. There are a bunch of neurons that are all talking to each other that are controlling, listening to sensory input and controlling motor output, with probably also some top-down information from the brain. So locust neurons function the same way that ours do. In fact, we've learned most of what we know about neurons from studying various invertebrate systems like the giant axon and the squid. And their brains are clusters of neurons like ours are too, but of course they're not nearly as complex, which makes them convenient for studying how sensory and motor systems, for example, talk to each other. And so the brain of insects have 3 lobes. You have the protocerebrum, the deuterocerebrum, and the tritocerebrum, all of which have specialized functions. The protocerebrum, which includes the optic lobe, which is pictured in B there, innervates the eyes and the ocelli, and we're going to learn about what the ocelli are, their sensory organs. The deuterocerebrum innervates the antenna, and the tritocerebrum innervates the foregut and the mouth parts. And of course, they're receiving other kinds of sensory information too from all the different regions of the body. The ganglia are clusters of neurons along the length of the thorax and the abdomen. They're named. You can see in the upper picture where their location is along the length of the locust. So you can see it's obviously starting at the brain, and then it's moving into the subesophageal ganglia, the prothoracic ganglia, the mesothoracic ganglia, the metathoracic ganglia, on down the abdomen. And then there's pictures in panel B of what kind of the cross section of each of those ganglia look like. And then the numbers are essentially nerves that are leaving the ganglia in order to go out into points in the body. Okay, so each muscle is controlled by 1 to 5 motor neurons. And these pictures are, of course, collections of individual muscle units and arranged into big flight muscles. And so you can see them pictured here, and they also have pictured on here some of the thoracic ganglia that are innervating and controlling the muscle contractions. And what we need then is for these muscles, the motor neurons that are controlling the muscles, we need them to fire at specific and appropriate phases of the wing cycle in order to produce that rhythmic wing movement. And you need them to be coordinated because you need the extensors to be contracting as the flexors are relaxing, and then you need for the wings to be in sync. And so you've got lots of different things happening. So in thinking about how you could produce a rhythmic pattern, some of the earlier hypotheses was that, well, you could have a series of reflexes that trigger each other to start when the wing is in a particular position. And this was called the chain reflex hypothesis. And it was thought that this might be kind of a simple explanation for how rhythmic motor patterns are produced. So an example here is that the brain senses that the wing is in the up position, and that sensation of the wing is up actually triggers the depressor muscle to contract and pull it down. And then when the wing is down, again, through proprioception, the brain senses that the wing is down, and that is what triggers the elevator muscle to contract and pull it back up. And so you can generate a cyclical rhythm through a series of reflexes. If that were the case, you would need to have the ability to know where the muscle positions and the wing position. So you need proprioceptive feedback. So this is sensory information from inside the body, as I mentioned before, and it gives information about a wing or a muscle's position, tension, movement, or direction, etc. And one of the, I want to say this is the only proprioceptor, but I don't actually know if that's true now that I'm wanting to say that. But stretch receptors are by far, I'd say, the most common proprioceptor in the body. I'm sure there, well, anyway. I'll have to look it up and see if there's other categories of proprioceptors. But stretch receptors are used throughout the body in lots and lots of different contexts. And what they're able to do is to determine the stretch of a muscle, a wing, a bladder, whatever it is in the body. And it's, again, just like the hair cell, we have a mechanical gate to the neuron. And so whatever the stretch receptor is either wrapped around or embedded inside the tissue, as that structure stretches, it actually physically pulls open cation channels that allow sodium to depolarize the neuron, and it fires action potentials. So you have a mechanical receptor, a mechanically gated receptor that causes the stretch receptor to fire action potentials. And the other thing that's convenient about them is the more you pull them, the more action potentials they fire. Essentially, the more excited they are, and they'll rapid fire action potentials. And so if a stretch receptor is just firing a couple of action potentials, it has a really low level, then you can, then the brain knows or perceives that organ or muscle is not stretched. But the more action potentials it fires, the more that structure is being stretched. And so there's a rate code for kind of how stretched the muscle is. And the brain can then perceive this as whatever it's attached to is in its kind of longest position or shortest position. So if we look in the locust system, we're going to talk about 3 different proprioceptors. And one of those is the wing hinge receptor. So where the wing attaches to the thorax, there's obviously connective tissues there to attach the wing to the exoskeleton. And so there are stretch receptors embedded in that connective tissue. And so when the wing is elevated, it stretches the receptor the most. And so you get more action potentials when the wing is elevated and fewer action potentials when the wing is depressed. So you can see in this trace on the bottom here that as the wing moves up and is at the kind of peak elevation, which is that dotted dashed line going across there, you can see that when that kind of reaches the peak, you get a big burst of activity in it, and it's really stretched until it then relaxes all the way back down. I think there's also maybe a motor neuron here on the bottom trace that's the depressor muscle. So you can see that shortly after the wing is in its kind of fully elevated position and that stretch receptor is firing off a bunch of action potentials to say, the wing is in its full position, then the depressor muscle fires and you get the motor neuron fires and you get the wing moving back down. The second kind of proprioceptor is called the tegula. And so in this picture of the locust head, you can see the box there, again, associated with the wing, of course. And in this one, it's stretched during the downstroke. So it fires action potentials when the wing is moving down. And so you can see the tegula output on the top of the top trace there. And it essentially fires just after the depressor muscle motor neuron fires. And so the depressor muscle is the one that's going to bring the wing back down. And so during that downstroke, you see a lot of activity in the tegula proprioceptors. And then we have the campaniform sensilla, which is actually integrated into the wing. And it's stretched the most when there's upward force or lift on the wing. So essentially what that means is that when you're pushing down the wing, you have an opposing upward force on the wing. That's what's generating the lift. And so again, similar to the tegula, as the wing is moving down and supinating, that's, you probably have heard the terms pronating and supinating in regards to human foot and knee structure. But you can also, anyway, you can talk about that with any limb or moving structure. It fires the most action potentials, again, when the wing is moving down. So it's going to give a different kind of information and specific to the wing than the tegula, but usually going to be in agreement for the most part. Okay, so what then subsequent experiments found is that flight and this kind of rhythmic firing of the muscles is not actually due to a chain reflex. And this was again Don Wilson, who was able to cut the sensory nerves from the wings going into those thoracic ganglia, which would then transfer the information to the brain or process it right there in the ganglia and control the motor output. And this is called deafferentation. In other words, you're removing the afferent information. You're removing the sensory information that's coming in from proprioceptors. And what happened is that the locust still flew with a typical wing beat pattern. Okay, so it still had the normal kind of forewing, hindwing offset that you saw, and his conclusion then was that afferent sensory information was not actually required for rhythmic muscle contraction. What was interesting though is that the wing beat frequency, so remember it's very consistent, the wing beat frequency in a flying locust, it actually dropped to half of normal. So essentially sensory information, the afferent information, is not required for the central pattern generator to generate rhythmic motor activity. But it certainly has the capacity to alter it and may be an important component for normal behavior, of course. So this was considered the first evidence for what he termed a central pattern generator, which is something that could create rhythmic output from the central nervous system. So CPGs are neural circuits that are capable of generating a rhythmic and patterned output in the absence of external rhythmic output. In other words, you don't need some external cue to say, like in the case of a reflex chain, to say, okay, now is when you fire. There's something about the neurons themselves that can cyclically create rhythmic activity. There's lots of different behaviors that use CPGs. So just about all forms of locomotion do. Walking, crawling, flying, running, swimming, talking, breathing, and chewing, defecating, urinating, and even ejaculating. All of those kinds of things are, at least the underlying pattern is generated by a central pattern generator in the central nervous system. But another feature of CPGs is that their output can be adjusted by internal and external feedback. So then came the search for what is the central pattern generator? What does it look like? Where is it located? What's controlling this rhythmic output to the motor neuron, to the muscle? So the first place that they looked is in the motor neuron. And if you put an electrode in the motor neuron, so remember when I say motor neuron, all I mean is that's the neuron that's directly stimulating the muscle. So you can have, you could have, central, I'm gonna say a central nervous system neuron, and then maybe a peripheral nervous system neuron, and that may or may not be the motor neuron. If it is the motor neuron, then it connects directly to the muscle. Okay, so you could, theoretically, you could have the motor neuron that's generating the rhythmicity of the muscle contractions, or you could have a neuron that's higher up in the control pathway, maybe somewhere in the central nervous system. So this highlights one problem. Well, so what you can see here on this trace is that you have really cyclical activity in the motor neuron, where you have the hind wing depressor and a forewing elevator here as examples. These are two different motor neurons. You can see they're numbered. You're going to see this a lot. These is motor neuron 128, that's controlling the hind wing depressor muscle, and then motor neuron 83, just below it, that's controlling the forewing elevator muscle. Both of them show really rhythmic output, and the rhythmic output makes sense in that they're offset, so you get elevation at a different time as depression. But one problem is that how can you tell if a rhythmic neuron is driven by a rhythmic circuit but doesn't itself create the rhythm? So in other words, your rhythmic neuron maybe 1 or 2 in this kind of lower, let me show you down here. So 1 and 2 below, so these two are just transmitting information from the central pattern generator neuron to the muscle. And so they're, yes, they're rhythmic, but they may not actually be generating the rhythm themselves. Alternatively, you could, if you're measuring a neuron and it's rhythmic, maybe it's the thing, it's one of the central things that is generating the rhythm. And in other words, is the actual central pattern generator. So if you're just investigating this and you put an electrode into a neuron and it shows rhythmicity, how do you know if it's a part of the CPG or if it's just in a chain that's driven by the CPG? And so it's showing rhythmicity. This is where reset experiments can come in. And so essentially you have the central pattern generator that's creating a cyclical motion or a cyclical pattern of activity. And so then, so are neuron 1 and neuron 2 and the muscle, right? They're all showing rhythmic activity because of the central pattern generator. So one of the ways that you can test whether or not a neuron that you're listening to is a CPG neuron or some other part of the chain is that you can shock it. You can give it a depolarization event in the middle of its rhythm. So you'd want to do it, you know, right, well, wherever, but either at the top or the bottom of a phase, right? So right there, you could reset it and see if you can get the timing of the rhythm to shift. Because if it's an actual part of the central pattern generator, then if you excite it, then at a different time than it's normally excited, you should shift when that rhythm is starting. If it's just downstream of the central pattern generator, so if you do it to that one, it'll affect it right then. But then in the next moment, when you're no longer shocking it, it'll be right back onto the original rhythm because the CPG neuron is still driving it. So you should be able to tell by these kind of shock experiments, these reset experiments, whether or not a rhythmic neuron is actually creating the cycle or is being driven by something different. So here's an example of a reset experiment. So at the top here, in all of these figures, you're going to see these boxes in the underneath the trace, and those boxes are essentially telling you when the rhythm is expected. Okay, so you have activation, and then you have a pause, and then you have activation, and then you have a pause, and then you have activation, and then you have a pause, and so that's the rhythm. So the boxes are always going to show what you would expect if the rhythm continues. In other words, so if the rhythm is normal. And then you're shocking it here. So here's this shock. And you're doing that in the middle of the cycle. And so essentially if it's a central pattern generator, then that should reset it to a new timing. And so you should see a difference between the previous phase and the new phase. If you don't see a difference, like in this upper version, you can see that the action potentials are still lined up with the expected period of activity. That suggests that that's not part of the CPG because it didn't change anything. It excited it right there in that moment when you actually added sodium ions and depolarized it yourself. But then it went right back to the original rhythm, suggesting that something else was the thing telling it when to fire. In these lower two versions, you do the same thing where you excite a rhythmic cell. This is obviously a different cell. And in this case, you can see that the activity shifts to be a little bit later. So that delayed the activity, and the fact that it changed it suggests that that cell might be important in generating the pattern. So you're in the CPG somewhere. This lower version, again, you excite the cell, and in this case, it actually shifted the activity a little sooner, and so that advanced the timing. Either way, when you change the timing, it suggests that this is you are stimulating a neuron that is part of the central pattern generator. And what these reset experiments did, they tested more than 80 motor neurons, and every single one of them failed the reset experiment. In other words, if you stimulate them in the middle of the cycle, it doesn't change the pattern. It doesn't change the rhythm of the motor output afterwards. And so what that means is that something else is driving the motor neurons to be rhythmic. It's not the motor neuron itself. They're just listening to a pattern generator somewhere else in the system. So the next proposition was that this must be the interneurons that are directly connecting to them within the central nervous system. So here's a picture here of one of the thoracic ganglia. And then you have a picture of an interneuron that's coming in from the central nervous system and then synapsing on a lot of different cells. So it has the ability to integrate a lot of different information from all of its targets, its dendrites, and then it's sending information to the motor neuron. And so here's, again, some more numbers here. So you can see that the motor neurons are designated motor neuron, MN, and the interneurons are IN. And so now you see some numbers here. And what scientists did next was they started investigating whether or not interneurons were driving the central pattern. And what they found is that indeed they've so far located 4, well, actually more than that. But here are 4 of kind of the first ones and some of the main ones. 4 interneurons that passed the reset test and were able to influence each other's patterned output as well. So interneuron 501, 301, 504, and 511, these are their connection patterns within the thoracic ganglia. And incidentally, when you see these kinds of patterns and pictures, the reason that you're able to create something like that is through what are called tracers. And so I put these at the bottom of the slide here. If you have located a cell body, one thing you can do is inject what's called an anterograde tracer into the cell body, and what that tracer does, so it's like a dye or something that can hitch a ride on proteins that travel down the axon. And so you'll actually see the trace, the dye, travel down the axon and then spread out to all of the cells that it synapses onto. So you can inject a cell and know which cells does that one talk to. In other words, which ones does its axon send information to? The opposite scenario is a retrograde tracer. And the way that those work is that you actually inject them into a synapse. So you're in a brain region and you want to know what other brain regions send information to this region where you're injecting the tracer, because those tracers go into the axon terminal and they travel backwards up to the cell body. And that's because there are also proteins. There's proteins that are carrying things down to the axon terminal. There's also proteins that carry things back to the cell body. So these tracers kind of hitch a ride and attach themselves to those proteins and get taken backwards or forwards up the axon. So that's pretty cool. Okay, so on the right side of this figure is a reset test of interneuron 501. And it's also showing the depressor muscle activity kind of along with it. So what you can see is that interneuron 501 becomes really active rhythmically. I'll just do it in blue, I guess it doesn't matter. So you can see this kind of rhythmic output of interneuron 501. And then the experimenter interrupts that with the depolarization event. And what it's showing now is the red bars is where we would expect 501 to start firing if it wasn't a central pattern generator. In other words, if we didn't disrupt the rhythm. And what you can see is that instead it's shifted to be later. And those dashed lines represent essentially how much it shifted. So, where it shifted to relative to the red bars where it should have. And so you can see that we successfully reset the rhythm, which means that 501 must be part of the central pattern generator, and it can therefore change the output and the rhythm of the central pattern generator. So it starts to get complicated really fast. And this is just showing how some of these different components of the central pattern generator can impact each other. So in the top one, you can see that interneuron 504 fires an action potential right there. And then shortly thereafter, ion 301 has an EPSP. So 504 excites 301. In the next trace, 301, then in turn, when it excites, it actually has an inhibitory postsynaptic effect on 511. 301 also, when it fires an action potential, excites 501. So you can see that 301 is impacting different cells differently. And so presumably it has GABA at the synapse with 511 in this one, and then GABA here and glutamate here, or some other excitatory neurotransmitter. Actually, I'm not sure what neurotransmitters these central pattern generators use. It's probably actually not glutamate, but it might be, who knows. I'm just giving you an example. And then when 501 fires, you get a little bit of an inhibitory postsynaptic potential in 301. The point here is that there's complicated relationships between all of these cells, that in any one of these 4 cells, if you did the reset test, it would change all the other ones' output, and it would change the pattern overall. So they're all able to influence each other. And presumably, the way that they do it can generate a rhythm. And we're going to look at a sub-circuit within all these cells in order to look at that theory. Even though it takes all of them to do what's done in the locust, we can understand it better if we look at a simplified circuit. So we're going to look at interneuron 301 and 501. And what we're going to see is that an oscillation of excitation and inhibition can create a recurring rhythm. And so we have just on the left-hand side in panel A, a wing beat cycle, and then the membrane potentials in 501 and 301. And what you can see, first of all, is that the peak activity in those 2 neurons are offset from each other, and they both peak essentially once during a wing beat cycle. So that already suggests that their rhythmicity is linked to the rhythmicity of the wing movement. Okay, so what we see is that interneuron 301 excites interneuron 501, but with a delay. The delay, it's not theoretically important, but what it's important for is creating a wing beat at the rate that you want it to be. So at that kind of 20 kilohertz. But anything, so you've got 301 that's exciting and causing an EPSP in 501. 501 actually then inhibits 301. So already now 301 fired an action potential, which excited 501, which turns off 301. So you've gotten kind of one cycle of activity there. When 301 is inhibited by 501, that means that you effectively turn off this transmission, and so you stop activating 501, and that stops the inhibition of 301, and you can start all over again. Okay, so you have excitation and inhibition that can lead to kind of a full circuit of turning on neurons and then turning off neurons, which allows the first neuron to turn back on again. So it can still be a little confusing, but it's pretty cool how one excitatory and one inhibitory synapse can effectively create a rhythmic pattern that could theoretically go forever. When you put all of the different circuits together, so this is the figure in Karoo. I'm going to show you another figure from a different paper next, but basically what you can see is that it gets complicated really fast, and that even in a simple system like a locust, there's still a lot going on, and it can be really hard to kind of map all of those different interactions. And there's still some blank boxes here where we know that there are neurons that are interneurons that are playing a role, but they hadn't been identified at that point. So the take-home points of this figure is that many interneurons are working together. There are excitatory and inhibitory connections within the network, and that's important that you have both. And they work together in order to rhythmically excite or inhibit the motor neurons in a very coordinated manner. You can see that these motor neurons on the right-hand side of the figure, the elevator that moves the wing up and then the depressor that moves the wing down, they have a lot of different interneurons that are synapsing on to that motor neuron. And some of them are excitatory and some of those are inhibitory. And so you've got kind of a lot of coordinated effort in order to generate the right pattern in the elevator and the depressor motor neurons. This is from a figure in a very recent paper. And obviously we've learned a lot in the last 20 years since Karoo was published. But again, as we see over and over again, the basics were really worked out, and a lot of them are present or still here in this figure. In other words, there haven't been any major mistakes that have been identified. But what I like about this figure is that one, it presents it so Karoo presented it kind of all stretched out, and so it doesn't look anything like it would look in the locust or in the thoracic ganglia that are controlling this rhythmic output. And in this paper, they tried to do it. It's more complicated to follow, right? You can't really follow this, but you can see the ganglia there. So you have the mesothoracic and the metathoracic ganglia, and how all of the locations of the interneurons within those ganglia and how many connections are traveling across ganglia to control these motor outputs. And also you can see visually that there are proprioceptive feedbacks happening. So the red circles are interneurons that receive information from the hindwing tegula. The green circles are interneurons that receive information from the forewing hinge. And so you have inputs where you have opportunity for sensory information to change the output of the central pattern generator. And that's where we're going to go next in video 3. So we're going to talk about proprioceptive and exteroceptive feedback next.